Okay, chapter 11. So we're going to be digging into neurophysiology. So this is our physiology chapter. The rest of the chapters are going to be more like neural anatomy. And so we have a little over a month to work on the nervous system, which is a good amount of time. Nervous system takes a little time, more so than the others. So let's go ahead and take a look at our nervous system. Basically, when we take a look at our nervous system, what we're looking at is a system that is basically a integration system. So the goal of the nervous system is to essentially regulate all the functions of the body, and we've already seen some pieces of it with the neuromuscular junction, but we want to kind of pull together the full spectrum of the nervous system. And we're going to start off first of all with the discussion of the tissues of the nervous system, and then we'll start to move more into regulation and physiology of the nervous system, and then follow that up with the anatomy of the nervous system as we move forward in the chapter. So the nervous tissue is composed of two dominant types of cells. You have neurons, which is the kingpin, if you will, of the nervous tissue, and then you have the glial cells, or the glia, the neuroglia. And so these two populations of cells form the bulk of the nervous tissue. Now, when you take a look at what we mean by some terminologies here, so for instance, the idea of a nerve is effectively just a bundle of axons. That's pretty much what a nerve is. So it's a bundle of axons. And so generally speaking, it's outside the brain and the spinal cord. So basically, it's more kind of peripheral in nature. And so we call them nerves and then tracts in the central nervous system. So they have slightly different terms. It's one thing you'll learn about the nervous system is synonymous structures in the central nervous system versus the peripheral nervous system have kind of slightly different ways of naming them, even though technically they're kind of roughly the same kind of structure in spirit. And so when you take a look at nerves, you have a couple of major types of nerves themselves. You have, first of all, what's called cranial nerves. These are called cranial nerves because they originate from the brain. You typically have 12 pairs of those. And they're dominant. They don't all work on the senses, but several of them, quite a few of them actually, are focused on your five senses. And then the rest are going to be spinal nerves. So they're originating from the spinal cord. And you have 31 pairs of those. Now, a couple of other types of working structures that we see in the nervous system are either collections of white matter or gray matter. And so another term we run into a lot is the ganglion. And a ganglion is an important thing to understand because a ganglion and a nucleus are kind of synonymous structures. A ganglion is basically a collection of cell bodies, neural cell bodies, that are typically outside the brain and the spinal cord. That is to say, in the periphery of the peripheral nervous system. And so these are processing areas, like processing stations that are outside the brain, the spinal cord. And then you have another term, which maybe several of you have run into, the concept of a plexus. Now, a plexus, all a plexus really means basically is a extensive network. It's kind of like a fishnet structure, and it's a generic structure. A neural plexus, in this case, is an extensive network of axons. And sometimes you'll have cell bodies in there, but typically it's outside of the central nervous system. That means it's the peripheral area. Just to let you know that you can also have a vascular plexus as well. That's basically a network of blood vessels. And then, of course, your glial cells, which basically are the supportive cells that essentially support the work and the activities of the neuron itself. Now, the important thing to understand is that these two rely on each other. The neurons are specialists. And so these ones are the highly specialized cells whose job it is to basically do the conduction, right? So they're sending the nerve impulse. And the glial cells are sort of like the support crew. They're like the entourage, if you will. You can think about it in a way, kind of like the neuron is sort of like the superstar, the one who's so specialized that they can't really even do basic functions anymore. If you take a look at somebody who's like really, really famous, they oftentimes have to have this sort of collection of like support people around them, folks who are kind of not necessarily advisors, but sort of like a support group around them. And their job is to do the basic things that the superstar can't do anymore, right? Like I'm thinking of somebody who's just like off the charts famous, somebody like a Michael Jackson in his day. You know, I mean, here's somebody, or like even here in Denver for you football fans, somebody who's like a John Elway, you know, somebody who's like so famous that they can't really function normally anymore. And so, like somebody like a Michael Jackson, for instance, can't really just go to the grocery store and buy groceries. You know, they can't really just go to the gas station and get gas or anything normal, you know, our normal errands and things like that we run on a daily basis. And so, in order to be able to get those sorts of things, they need to hire people to do that for them. And that's kind of their support group, right? So, there was somebody who was doing all those menial errands for these superstars. And that's kind of what the glial cells are doing. So the neurons are so specialized, they're so sort of famous, if you will, that they can't even function half the time normally as a cell, and they need other cells to do some of those functions for them. And so, an important piece, because you have this sort of support network between the neuron and the glial cells, it's important to maintain your homeostasis, right? So this is one of the big things that you have to have homeostasis maintained for the nervous tissue, and the nervous tissue has to maintain homeostasis for the rest of the body, right? So when you take a look at your nervous system, you're taking a look at the one whose job it is to regulate and to coordinate homeostasis and maintaining that balance. Their job is also to take in all the sensory input that you're collecting from the outside world and to constantly monitor both external stimuli and internal stimuli. So the external stimuli, which is the stuff that's coming from your environment, the internal stimuli, which is the stuff that's coming from within you. So your nervous system is also having to monitor the things that are happening inside your body, the things you're not even aware of, so that basically everything is maintained in a constant state of balance, that is to say, homeostasis. So once they receive this information, then it's their job to integrate that information. You do that in your brain, your spinal cord for the most part. And then they have to come up with some sort of a decision based on that information. That's your response. And then the responses will then go to targets, muscles and glands, for instance, in order to act accordingly based on the information that you just received. And so this is the way that we're able to maintain just basic function. So if we're talking about just basic consciousness, like when to be asleep and when not to be asleep, thinking, the thoughts that we think, the memories that we have, the emotions that we have. So all of these are coordinated and kind of integrated responses of these inputs that are coming in. And so you can basically break your nervous system, and I've already mentioned a little bit a couple of times, into two major components. You have the central nervous system, which is basically your brain and your spinal cord, and then your peripheral nervous system, which is pretty much everything else. So it's the sensory receptors and the nerves and all those organs that are basically spread out throughout the rest of your body. So when you take a look at your central nervous system, it's going to be limited pretty much to your brain and spinal cord, and that's pretty much it, right? But there's a lot going on there, and that's going to keep us more than busy. Now, the peripheral nervous system has a lot more stuff in it, if you will, more structures in it. For instance, they've got receptors in the peripheral nervous system. They got nerves in there. They got ganglia and plexuses. So they got all these different kinds of wiring strategies associated with the peripheral nervous system. And you can break the peripheral nervous system into what's called divisions, right? So the first one you can create is what's called the sensory division, and you have the motor division. So you're either bringing information in from the external environment or you are acting on that information in response to that what's coming in from the environment. This also will basically determine the direction of the signal. So for instance, afferent is oftentimes the more technical term for sensory input. So the afferent system is heading toward the brain, having collected sensory information, and the efferent is basically moving away from the brain, having received the response based on that sensory information. It's going through the efferent, and then from the efferent, it's going to then go into a branch of the PNS called the somatic branch. And so the somatic branch is typically the one we associate with the control of our skeletal muscles, which will then basically contract. And so we learned about this in chapter 9. So that neuromuscular junction was all a component of the somatic system. Now, the somatic system is absolutely voluntary. So you can choose whether or not to move your skeletal muscles. You have complete control of that one. However, there is a second branch of the efferent system that is basically taking the response from the motor division, and that is the autonomic. So this is the stuff that's on autopilot. It's the involuntary responses. And you basically will split the autonomic system into two different subdivisions. One, which is your sympathetic division, which is better known as your fight or flight mechanism. This is your stress response. And generally speaking, the sympathetic division is going to be targeting lots of different structures, your cardiac muscle, your smooth muscle, and your glands. But this is basically getting you ready for physical activity or like running for your life, right? Now, the other subdivision of the autonomic is the parasympathetic. So this is basically your rest and digest subdivision. It's also going to be targeting the cardiac, the smooth, and the glands. But in this case, what's happening is it's regulating your resting functions. So this is basically the one that's in play when you are at rest. So many of you probably are at rest right now, and so you're probably in parasympathetic mode. However, on Tuesday, last Tuesday, when I let you guys into the room and I started handing out all the exams, all of a sudden, many of you probably moved into the sympathetic division. Your stress level started going up. And so you kind of started switching into the other side of the autonomic nervous system. So those are the major organization of your nervous system. So here's kind of what it looks like in pictures. So you can see your central nervous system, which is composed of your brain and your spinal cord. We'll have a lot to say about that. And then, of course, your peripheral nervous system is associated with all the nerves that are coming off of the spinal cord and innervating your body, right? So, for instance, the nerves going to your head and face, your thorax, your limbs, your abdomen, your legs, so forth. And then if you take a look at these little swellings, like for instance, right here, these little beads, if you will, those are ganglia. Those are little piles of cell bodies that are essentially processing stations in the peripheral nervous system. It allows you to be able to coordinate very complex sensory signals and be able to kind of sort through them and to pass them on in a meaningful way so that your brain doesn't have to sort through all of that mess. You could imagine what a nightmare the postal system would be if everybody had to go to the post office and basically dig through all the mail and find their own names and their own addresses. So the mail, sorting it out by zip code and by address and by division, helps to make the delivery much, much more efficient. So you can think of the substations of the post office as sort of these ganglia, if you will. They're processing stations designed to sort of bring in lots of complex pieces of information, to sort them all out and to basically move them out in an orderly, efficient manner so that they can be delivered easily and in a timely fashion. That's exactly what ganglia are doing. Okay. So when you take a look at sort of what they're doing, you can see that basically you have two major divisions. You have your motor and your sensory division. So your sensory division typically is peripheral in nature, and your motor division is also peripheral in nature. And so at first, you start off with your sensory inputs. So you're basically either seeing something, hearing something, tasting something, things like that. That is going to be picked up by sensory receptors and then sent up to the spinal cord, where the brain will make some sense out of it and it'll send it into the motor division for its response. And so you'll either send it back down and it'll go through either the somatic nervous system, where you will contract your skeletal muscles, or it'll go through your autonomic nervous system, where it will either activate the sympathetic nervous system or the parasympathetic, depending on the nature of the message that the CNS is sending out. So this is what your system looks like, broken down by division. Okay. So the sensory, the afferent division, if you will. So basically, the goal of the sensory is to send action potentials from the receptors. So you got your sensory receptors out there, and it's going to be moving it in toward the central nervous system. And so when you take a look at receptors, there's a lot of different types of receptors throughout your body. Typically, they can be nerve endings. They can actually be specialized types of structures, specialized cells that are basically doing the same thing. They're detecting external and internal stimuli, and they're sending that information, what they're learning, they send that to the brain, into the spinal cord. And then the motor, the efferent, will basically transmit action potentials from the central nervous system to your effectors, which typically are muscles and glands. So then from our motor division, as we break into the somatic versus the autonomic, the somatic basically will go directly to the skeletal muscles. So as a matter of fact, we've already done a lot of work, believe it or not, on the nervous system in chapter 9. So when we were going through all of that work on the neuromuscular junction, a lot of that was us studying the somatic nervous system, and especially the contact point between the nervous system and the skeletal muscles, the terminal target, where we actually get contraction. And so this is the voluntary piece, and it's typically a single neuron system. So it's basically wiring all of your muscles with the appropriate nervous regulation that's necessary for correct muscle contraction. Now, the autonomic nervous system is critical, the ANS, because this is basically the one. So when you get into AMP2, you're going to be going through all the core systems. And one of the things you learn about each core system is that they are all controlled by two major systems in your body. You always have neural control, the nervous system, and they also have endocrine control, the endocrine system. So typically you start off AMP2 with a discussion of the endocrine system. But a lot of these visceral organ systems are governed and driven by the autonomic nervous system. And so typically this is signals coming from the central nervous system. It's going to either the smooth muscle, say for instance in your digestive tract, or cardiac muscle in your heart, or glands. So these are all things that are automatic. So they're subconscious and involuntary in nature. You don't have to think about these in order to get them to work. They just kind of work on their own, which is really what you want, right? Because after all, you don't want to have to keep telling your diaphragm to contract every time you want to take a breath. That would be too labor intensive. Generally speaking, when you take a look at the autonomic nervous system as opposed to the somatic nervous system, instead of just having a single neuron system, you've got two neurons in your autonomic system. Basically, they're either called preganglionic or postganglionic. So normally, the first piece is going to go from your central nervous system to a relay station, which is your ganglion. And then from the ganglion, it's going to go to your second it's going to go to the second destination, which is your effector. So it goes from CNS to the ganglion, and then from the ganglion to the effector. So you have basically two neurons. So this is neuron number one. This is what's called the preganglionic. And this is neuron number two. This one is postganglionic. And so that's common between the subdivisions of the autonomic nervous system, or the divisions of the autonomic nervous system, which are the sympathetic, which is going to be your stress response, and the parasympathetic, which is basically associated with just at-rest functions, digestion, things like that nature. You also have another subdivision, which is actually kind of a specialist subdivision called the enteric nervous system, or the ENS. And this basically is governed by what's called a series of plexuses, or these are networks of neurons in this case. You find them in the wall of your digestive tract, especially the wall of your stomach, and these basically will regulate digestive activity. So think of it as the ENS has basically had the digestive functionality delegated to it. So generally speaking, your central nervous system is in control of both sympathetic and parasympathetic, but when it comes to the digestive system, there's so many things going on in the digestive system. There's so many like hormones that need to be secreted, and there's muscles that need to be contracted, and there's timing situations associated with this, and you have to be able to move your food bolus through at very specific times under very specific circumstances. So because there's so much regulation, there's so many things going on in the digestive system, this autonomic nervous system kind of sort of hands off or delegates the control of that digestive system to the ENS, the enteric nervous system. Okay, so let's go ahead and review our neurons, kind of like we did in our cell tissues chapter, just kind of refresh your guys' memory. Remember, a neuron is our conductive, excitable cell, right? And so we have a couple of major pieces to our neuron that are important. First of all, the cell body, or the soma. Soma actually means body. That's basically essentially what the neurons are doing, is they're doing a lot of protein synthesis. For instance, these guys are the ones making the neurotransmitters, and they're doing basic cell maintenance and things of that nature, keeping the cell healthy and alive. And they create a special type of rough ER. So they have rough ER in there. You guys might have remembered rough ER from your general bio. Right, rough ER is the one that's the organelle in the cell that is studded with ribosomes, and it's responsible for the synthesis of secreted proteins, lysosomal proteins, and membrane proteins. And so in the cell body of a neuron, you've got a large cluster, or a large amount of these rough ER, and so they refer to those as Nissl bodies. So you can actually see them cytologically. Of course, they were named Nissl bodies before we realized what they were, but now we know they're rough ER. Another major piece of the neuron are the dendrites, right? So the dendrites are small membranous processes that sort of stick off of the body of the neuron itself, and their job is to receive information coming in to the neuron, typically from other neurons, whether sensory or not. But they'll usually have sensory receptors on them. They tend to be fairly short, and there happens usually there's a lot of them, and so they will oftentimes have these little projections on them called dendritic spines, like these little spiny looking structures. And their job is to basically conduct currents toward the cell body. So they're moving stuff in toward the cell body. So the cell body is collecting information using these dendrites to do that. And then, of course, the last and probably the major of these structures is going to be the axon. So the axon basically arises from an area of the cell body called the axon hillock. It looks kind of like the top of a funnel. And so this is basically where you get kind of summation of all of these incoming signals, and it represents the initial segment associated with depolarization. And we oftentimes refer to this as the trigger zone, where the action potentials are generated, and then they start to move down the axon. So if you guys remember when I did that slide, when we were talking about the control of the physiology, we were talking about the voltage gates and the rolling depolarization of the voltage gates. That initial generation of action potential is happening in this area, in this axon hillock, this trigger zone area. Now, the axoplasm is basically the fluid inside the axon. And the axolemma is the membrane of the axon. Remember when I said that the muscle people kind of had their own names for everything? Sarcolemma was the plasma membrane of the muscle cell and things like that. Kind of it's similar with neuron people, right? So axoplasm is the fluid inside the axon, and then the membrane of the axon, which essentially is the plasma membrane, is the axolemma. And this will cover the axon as long as it goes, until it terminates into the presynaptic terminal. That's the end piece. And that is where we will see the synaptic vesicles and the synaptic cleft. So this is where the synaptic vesicles are being held that are full of neurotransmitter. And so this is basically what our neuron looks like. So here you can see the dendrites, which are all these little small fibers basically coming into the cell body. So bringing in lots of different incoming messages into the cell body. You can see the Nissl bodies, which are these large clusterings of rough ER. And then down here, this little top part looks like an open end of a funnel. This is where your axon hillock is. And the initial segment that gets depolarized is right on down here, right at the very beginning of the axon. And so you can see the axon basically will be this long process, the longest of them all. So this is what an actual axon looks like. You can see very long, wispy process that's on its way to whatever its target is. And then you can see the axolemma is basically the covering of the axon itself. It's, if you look inside, it's not pointing to the myelin sheath, it's pointing to the actual plasma membrane of the axon. So that's the axolemma. On top of the axolemma is the myelin sheath, which in this case for a peripheral nerve is created by the Schwann cells. And you can see you have different Schwann cells that kind of wrap around the axon, kind of like insulation wire, like insulation on a copper wire. And that basically insulates this electrical signal as it goes down the axon itself. And then it keeps going down until you get to your presynaptic terminals, where you have lots of these little branches called telodendria. And these are those little feet that basically will attach to muscles, muscle fiber bundles. Now, a lot of times you also have kind of a little offshoot of the axon. This is what's referred to as a collateral axon. So this kind of helps you to further connect to different neighboring neurons to coordinate your response. So this is basically what a neuron looks. This is a classic motor neuron. That's what a classic motor neuron looks like. You can see all these dendrites that are coming in the cell body, which is this big guy right here. There's your nucleus. And then here's your axon hillock. And then your axon. Now, these little purple circles here are the nuclei of glial cells, where these are the support cells for the neurons. So that's kind of what your neural tissue looks like. And that's the functional pieces of your neuron. Now, one thing I do want to mention, because we're going to be talking about it later when we talk about propagation of an action potential, is in between these wrappings of Schwann cells, you're going to notice these little openings, open areas here of exposed axon. Those are intentional. Those are what we refer to as the nodes of Ranvier, or the axonal nodes. And so this may look familiar to you. This is basically a neuromuscular junction here. So you can see you've got the axon branch coming out. This is that little terminal foot where it interfaces with the muscle fiber itself. So when you take a look at what's inside the axon, then inside the axon, the axoplasm, which is just fluid, is basically going to be moving proteins, mitochondria, synaptic vesicles. Basically, it's going to go from the cell body toward the terminals. This is what's called anterograde movement. And so this is your supply for growth and repair and renewal. So you have cytoskeletal proteins moving toward the axon terminal. You got mitochondria that are moving toward the axon terminal. You got synaptic vesicles that are moving along the axon terminal. Now, you also have movement coming the opposite direction, right? So movement that's going toward the cell body, the return trip, is what's called retrograde, right? So retro to return. And so this includes, for instance, if you have damaged organelles, you got to bring them back. If you're recycling your plasma membrane, you got to bring that back. If you take in things by endocytosis at your axon terminals, you bring that on back. And so this is kind of an important piece as you kind of bring it back to the cell body for processing. So you have traffic basically going back and forth. Now, this is kind of important. So this is basically how the viruses for rabies and herpes can move. They basically move through axons through this retrograde type of movement. Now, if you take a look at it, if you kind of blow it up here, let me see if we actually have a blow up of this one, because if not, I want to draw it just to sort of draw it, make it clear. Okay, so this is basically our axon. I'm going to blow it up. Ready? Here's our axon. This is our axolemma. And so basically what you have laid down, and so it's filled with axoplasm, but basically what you have laid down in the axon is these long protein filaments called neurofilaments. And think of these like microtubules. So basically it's on these microtubules that you're going to see these motor proteins essentially walking some sort of a vesicle out toward the axon terminus. This direction is your axon terminus. Or they'll be walking some sort of a protein. And they'll be moving toward the axon terminus. Or they may even be walking, well, it's kind of big, they'll probably need a couple of them, a mitochondrion heading out toward the axon terminus. So they're basically moving stuff to the axon terminus. Now, where does this vesicle, what's inside this vesicle? The vesicle contains your neurotransmitter, right? So that has your neurotransmitter in it. And so those are those synaptic vesicles that are at the axon terminus that are just hanging out, waiting for the action potential to arrive. Where do those come from? Well, it's simple. The cell body makes the neurotransmitters, oftentimes, especially if they're protein neurotransmitters. But they'll make the neurotransmitters, pack them in vesicles, and walk them down to the axon terminus. Now, you also have, this is the anterograde direction, and then the retrograde would be the return reaction. So now basically you have these motor proteins that are taking, let's say, damaged organelles, and they're moving it back to the cell body. So that's your retrograde. So you're taking stuff that you collect at your axon terminus and bringing it back to the cell body, and they're moving things out to the axon terminus supplies that are needed. So there's like a back and forth going on. Okay, so that's our basic neuron. Now that we've got our basic neuron down in a lot more detail than we had in our tissues chapter, which is the reason why we kind of did a little bit more of a light touch in the tissues chapter, because we're going to go into it deep in the neural chapter. Let's take a look at some of the classifications and the functions of neurons, specific types of neurons. So we can classify them in a couple of ways. First of all, we can either classify neurons by their function. For instance, we can basically say whether they are sensory or afferent in nature, so they're going to be sending their action potentials toward the CNS, or whether they're efferent or motor, in which case they're going away from the CNS, or whether they're interneurons, that is to say they're within the CNS typically, but essentially they're a relay neuron going from one neuron to another. So they're not really, they're just kind of like passing on whatever the stimulation is, or the message is. We can also classify them structurally based on their dendrite numbers. For instance, we have what's called a multipolar neuron, which basically is a lot of neurons in the CNS, motor neurons as well. We have lots of dendrites coming into the cell body. You have what's called bipolar. You'll find this in the sensory cells in the retina, bipolar cells of the eye, and also you'll find them in the nasal cavity as well. And pseudo-unipolar, which is what it says it is. Pseudo means fake, unipolar. So this has a single process that essentially divides into two branches. So it looks like a single process, but it's actually two. And so part of it will go to the periphery that has kind of a dendrite-like kind of sensory receptor. But we'll take a look at what those look like. And the fourth one is what's called anaxonic. That is to say, it has no axons, only dendrites. And oftentimes we'll find these in tight clusters of neurons that are oftentimes found in the brain and in dense areas of the retina. Basically, they're only communicating very small types of voltages, graded potentials between each other. And so these are more like for coordination. So they do mostly coordination. So oftentimes the reason why you would have an axon is because you want to propagate your message across distance. But if you're not really moving your message across distance and all you really need to do is just talk to the cell that's right next to you, then all you really need is a dendrite connection. So you don't really need an axon, you just need dendrites. So as a result, what happens is these tend to be in the brain, because in your brain, what you're doing is you're interconnecting your neurons that are typically in close proximity to each other, but you're kind of connecting these neurons together in very specific and alternate pathways. And then creating these different alternate pathways is a way to create kind of a sequential pathway that is translated into a piece of information or like a memory of sorts. So here's kind of what they look like. So you can see your multipolar basically has lots of dendrites, so multiple poles that are basically connecting to the cell body. A bipolar has two main poles. So here's the first one, the dendrite on one side and the axon on the other. A pseudo-unipolar looks like there's only a single pole that's attached to the cell body, when in reality it bifurcates into really two types of poles. So it's kind of like It's kind of like a bipolar, only instead of the cell body being in the middle of the road, it's actually kind of pulled off like on an off ramp or something like that. And so this allows you to be able to sort of monitor the conversation between, say, for instance, two pockets of dendrites, either dendrites that are coming in versus your terminus. And so it kind of puts you basically in the middle of the communication. So it's kind of a slightly different organization. And then the anaxonic neuron basically has nothing but dendrites, because you're receiving information and you can also use those dendrites to send information. So this is like for high coordination of messages and processing of messages. Okay. So those are the major structural types of neurons that we have. And typically you see mostly multipolar or anaxonic neurons. Usually you're in very specific tissue types if you start getting into pseudo-unipolar or bipolar neurons. Okay, so now that we've talked about the neurons, let's go ahead and start talking about the glial cells. So the first glial cell, there's 5 of them that we're going to talk about, is the astrocytes. The astrocytes are kind of like the nurse cells. So they tend to be very star-shaped in process. So here you can see an astrocyte, which basically has these sort of flat, sort of flipper-like extensions that attach to an interface with the cell body of a neuron. And then another appendage will basically wrap itself around a capillary and a blood vessel. So these foot processes basically are characteristic of the astrocytes. And so as a result, they are basically interfacing with both the neuron cell body and the capillary. And so they will cover the blood vessels and the inner layer of the meninges called the pia mater. They'll regulate basically the fluid of the brain, that's composition. And this is basically what we oftentimes refer to when we're talking about the blood-brain barrier. Has anybody ever heard of the blood-brain barrier? Sometimes when you hear the term blood-brain barrier, especially when you hear people talking about it, doctors and researchers, it sounds a little abstract, right? I mean, it doesn't really sound obvious what it is. And this is what it is. This basically is the blood-brain barrier. Why? Because unlike most cells, the neuron isn't actually receiving nutrients and oxygen directly from the capillary, which is kind of like different than what other tissue cells do. Other tissue cells are directly interfacing with the capillaries and bringing in their oxygen and nutrients directly from the capillaries, typically by diffusion. But neurons don't do that. So neurons actually have astrocytes. All the nutrients and the oxygen will be picked up by the astrocyte. It'll be moved into the astrocyte and then delivered with the other foot process selectively to the actual body of the neuron. So let's kind of further think about this for a second and the nature of the way neurons work. Neurons, we said, are highly specialized, right? They're so specialized that they've lost the capacity to do basic functions, like including feeding themselves. So you can think of the neuron in a lot of ways like a baby, right? I mean, when a baby is little, do they eat? Do they feed themselves? How do the babies get food when they're really little? Yeah, it all comes through mom. Dad too, but usually dad, unless he's like a Mr. Mom kind of guy, is let's face it, dad's more like the entertainment division, although mom has a pretty good time with it too, right? But basically, when you take a look at it, the parents are taking a look at every single scrap of food that goes into that baby's mouth, right? Now, oftentimes, you take a look at it as a parent, you're not just basically picking food up off the shelf. You're really picky, right? You're looking at nutrients for your baby. You're looking at mixing in vegetables and different types of food types to expose your baby to different flavors and getting them different nutrients. And so you're like the nutritionist for your baby. And so everything that goes into that baby's mouth is filtered and selectively processed through the parent. So in a lot of ways, the parent is like the astrocyte and the baby is like the neuron. And so that is like what the blood-brain barrier is. So things that are in the blood don't just get to have free contact with the neuron. It's all going to go through and processed and screened by the astrocyte first. And so this is the controller of who gets to come in to the CNS. And so oftentimes when you have compromises in this, you have like reactive astrocytosis. This is basically where you have an injury site, kind of walls off that injury site to kind of cordon off or kind of contain the inflammation, because inflammation can spread. You don't want to get inflammation, widespread inflammation in your nervous system. Nothing good's going to come from that. And so this is one way to kind of wall that off a little bit. And oftentimes the astrocytes are important, especially in axon healing and neural healing. Now, the astrocytes, if they're around, they can promote the development of synapses. They can basically help stimulate connections and things of that nature. They can also help a little bit with damaged axon recovery and things like that, neuron recovery to a limited extent. By and large, typically, unlike most cells in your body, neurons generally are not replaced. Neurons are typically lost. Now, for the most part, as a general rule, neurons don't divide. And so as a result, if you were to damage a neuron, it's basically a permanent loss. So it's not like you can just basically make a new neuron and plug it in there, because neurons generally do not divide. And so that makes them kind of a little bit different than most tissues in the body. Now, they have, and we have done research where we've been able to get neurons to divide, and they have a very limited kind of a division capacity, but nothing that's even remotely close to a functional level to where we could say like, oh, hey, this is a therapeutic treatment, say for instance, Alzheimer's, where we're actually losing neural activity or functional neuron connections. So generally speaking, whenever you get neurodegenerative disorders, oftentimes, not all the time, but oftentimes, it's associated with neural death. So the death of a neuron, which is a permanent death, and it's a permanent loss in the brain. So whatever connections were being held up by that neuron will be lost. And so that's kind of important because when you take a look at astrocytes, there is a little bit of capacity for astrocytes to support neural development in terms of synaptic connections and limited healing. But generally speaking, nerves are difficult, and you got to get lucky for nerves to heal properly. And oftentimes in an injury, you're often not lucky. So you just end up having to deal with some sort of a neural pathology for the rest of your life. Like if you injure your hand, sometimes you're just going to live with numbness in your hand because the nerves don't heal back. That happens quite frequently. Another group of cells, the next group of glial cells, what's called the ependymal cells. And so these guys are important because they will line the ventricles of the brain. Now, the ventricles are basically open chambers in the brain, and the spinal cord, which is the central canal. So these will line these open chambers in the brain and the central canal, the spinal cord. And so there's versions of this in a structure that's called the choroid plexus. And the choroid plexus is found in one of the ventricles. And so typically they are ciliated in nature. So here's an example of what the ependymal cells will look like. So here's a line of the cells lining the ventricles of the brain. And the cilia will move back and forth to help circulate this cerebral spinal fluid. And on the choroid plexus, you have a specialized group of these ependymal cells. And their job is to actually make the cerebral spinal fluid. So the choroid plexus is the one that actually produces the cerebral spinal fluid. And it actually makes it. So it secretes that cerebral spinal fluid into the ventricle. And then the cilia help move the fluid through the cavities, the ventricles of the brain. And so these are going to be important because if you don't have cerebral spinal fluid, then you don't have the necessary fluid to sort of support your central nervous system. So you kind of have your own special fluid in your central nervous system that's a little bit different than the other fluids. So those are the ependymal cells, very important. Choroid plexus is going to be the main structure that we see with ependymal cells. Now, the third group of glial cells is going to be the microglia. These guys are macrophages. So they basically have kind of an immune response. They have sort of an immune role. In the CNS. So they're phagocytic. That means they'll take a pathogen, they'll gobble it up. And so they can respond to inflammation. So if inflammation is occurring, they can respond to that. They can phagocytize stuff. So they can go after microorganisms, gobble them up like bacteria. They can go after dead tissue, necrotic tissue, so stuff that dies, debris, very similar to the way normal macrophages work. And anything that's foreign in that central nervous system, they can gobble up. So this is kind of like your hired bodyguard. Right? Every famous person has to have bodyguards. I'm certain Michael Jackson had his bodyguards, right? But that's a member of your entourage. So you got the people who go out and do the grocery shopping for you. You got the bodyguards who basically take care of anybody who's got nefarious intentions and things like that. And so here's an example of a microglia. You can see it's a very kind of a stringy dendritic kind of a looking structure. But it's important. Why? Because the reason why you have microglia here is because of that blood-brain barrier, right? The astrocytes. Normal immune cells don't have access to the brain or to your neural tissue because they're in the blood, but they have to get out of the blood and they have to get into the tissues to be able to do their controlling. And so since they can't cross the blood-brain barrier, then they're kind of trapped in there. So that basically means that your nervous tissue is vulnerable to potential attack. If a pathogen gets into your nervous tissue, it's kind of like getting into an area where there is no law. They can do whatever they want. So having your microglia is like having your hired bodyguard, right? If you want to think in terms of like the Roman emperor, right? The Roman emperor had his own military garrison called the Praetorian Guard, whose job originally was to protect the emperor when he was out in public from assassination and attack. Very similar to the way our Secret Service works for the president, right? Only in Rome, the Praetorian Guard actually became more of the organized assassinators of the emperor rather than the protectors of him. But that was their original job. Okay. So the fourth type of glial cell we're going to see is what's called the oligodendrocyte. So an oligodendrocyte is important because what it's going to do is it's going to create what's called a myelin sheath. And so basically, it forms a cytoplasmic extension around the axon. It just basically wraps itself, its membrane, around and around and around and around the axon. And so what you see are these just multiple, they kind of, if you take a look at the oligodendrocyte, so here's its like little, here's the cell, and it's got this little extension. So what it does basically is it just kind of takes this and kind of wraps itself around the axon and just keeps on wrapping itself around the axon, around and around and around and around, multiple layers around that axon. And that creates this very strong kind of lipidy, right? The myelin sheath is lipid due to the phospholipid bilayer. And as a result, it's non-conductive, right? So it's like an insulation. So it basically forms insulation around these axons, but the oligodendrocytes are basically only found in the central nervous system. So these will cover myelinated axons and myelinated neurons, right? So there are a lot of central nervous system neurons, however, that do not have myelin, so they're unmyelinated. Now, the fifth type of glial cell we'll see. Now, these guys are going to be in the peripheral nervous system. So far, everything previous to this particular slide has all been in the central nervous system. Now, we're in the PNS, the central nervous system. The first one we're going to be running into are the Schwann cells. Basically, these guys are synonymous with the oligodendrocytes. So they do the same thing. They basically will wrap their membrane around an axon, creating a myelin sheath. And so this creates that lipidy kind of phospholipid sheath around it, which creates that myelin sheath, which again is non-conductive. So it's basically the same thing as the oligodendrocyte, only in the peripheral nervous system. That insulation is formed by the Schwann cells. In addition to this, you also see what's called satellite cells. So this is the fifth type, I forget what we're on, sixth type of glial cell, also peripheral system. This will actually cover the neuron cell bodies, and generally they'll be focused on sensory and autonomic ganglia, and their job is to sort of act like astrocytes. In support of the neuron, or in support of the cell body. So their job is to basically provide support, nutrients, protection for heavy metal poisoning. So these guys will screen out heavy metals. So they're kind of operating in a lot of ways very similar to the astrocytes. So if I want to put a CP on these guys, I would say CP to oligodendrocytes. And this one CP to astrocytes. So this is an example of two structures that have synonymous roles, but in the central nervous system versus the peripheral nervous system. Now, when we take a look at our axons, then that kind of sets us up for our two major types of axons, right? So we have our myelinated axons, which basically has this myelin sheath that protects and insulates the axons so that they can have fast transmission. They can also help guide axon repair. So if you damage a neuron, your axon, if your myelin sheath is still intact, it can actually help to guide the repair of axons to a certain degree. They oftentimes are not continuous. That means they have gaps, right? These gaps we mentioned before are the nodes of Ranvier, and this is what causes white matter. So the reason why white matter is white because of the lipids, because that lipid bilayer is whitish in nature. Now, if you were to degenerate the myelin sheath, then what you're going to get is discoordinated control. So, for instance, multiple sclerosis is myelin deterioration. And in some cases of diabetes, you can also get myelin deterioration, which can create discoordinated either muscular control or communication between neurons. Now, the unmyelinated axons typically are not wrapped. They'll kind of be hanging out in little pockets of Schwann cells and oligodendrocytes, but they're not wrapped. That's the important thing around the axon. And so they have no myelin sheath. And so as a result, they don't have that whitish appearance, so they're gray. So if you don't have that white myelin, then you're not going to have that whitish appearance. And this is kind of what it looks like. So here is your myelinated axon. You can see how you have the nucleus of your Schwann cell. In this particular case, it's wrapping itself around multiple times around that axon. And so that creates the insulation. And so where the two cells come together, you have that little divot or that little pit. That's the node of Ranvier. It's actually the junction between two Schwann cells. And then the other Schwann cell will pick up and wrap around its segment. Now, in the unmyelinateds, in this case, you're going to have your individual axons, but then now it's not like they don't have some covering. They're not just raw axons out there, but basically this is kind of where the Schwann cells kind of reside. So here's your first Schwann cell, and you can see how they're kind of in these little pockets, basically these invaginations, if you will. They're kind of gobbled up by the Schwann cells, but they're not multiply wrapped. So they still have a little bit of coverage, but there's much more exposure. So they're not as tightly wrapped or as tightly insulated as the axon that is wrapped up in a myelin sheath. And so in both cases, because you have the need to protect the axon, you're going to have a Schwann cell or an oligodendrocyte covering the axon. It's just a matter of how, right? If you have heavy insulation, the myelin sheath, then you're going to have really, really tight protection, good transmission of that electrical signal. Oftentimes, this is what you want if you need to propagate that signal across large distances. And then in the unmyelinated sheath, it's going to be not heavily wrapped. It'll be invaginated, so it'll have a little bit of cover, but there'll still be a lot of exposure. So this is ideal for a situation where you're just going to be transmitting your action potential just to a local or a nearby cell. So you don't need that heavy insulation because you're not going to be going very far. So you can just basically spark it over to your next door neighbor. And that's the extent of your communication. So since we are at our witching hour, this is a good topical shift to start moving into the response of nervous tissue to injury, and then we'll start digging into the physiology of the nervous system.